السلام عليكم ورحمة الله وبركاته. معكم إباء زيادنة من قسم الفسيولوجي. I will start our lectures on reproductive systems. We will start with male reproductive system or physiology of the male reproductive system, and then we'll continue with the female reproductive physiology. So in this lecture, and this is chapter 81 from your textbook Guyton and Hall, our objectives of this lecture is to explain the regulation of the male reproductive function, and I explain the spermatogenesis, the process of sperm's formation, as well as the main functions of the male reproductive organs. We will also be able to describe synthesis, secretion, and metabolism of the hormone testosterone, as well as its various effects in the male. We also will identify some of the abnormalities of the male reproductive function, just to emphasize the role of the components of the reproductive system in male. So now let's identify the main organs of the male reproductive system. As you can see here from the figure, the main organ of the male reproductive system is the testis, which is the gonad of the male reproductive system. لأنه بيتم في تكوين السبيرمز بعملية أو process that is called spermatogenesis. And the testis is a coiled organ. It is composed of 900 coiled tubules. طول كل واحدة منهم about 0.5 meter. Then the content in the seminiferous tubules will be transported into another coiled system or organ, which is called epididymis. And the epididymis is also a tubular system that is 6 meters in length. Content of the epididymis will go through or empty in the vas deferens. Vas deferens, as you can see here in the upper figure, it links the testis, epididymis with the internal and external parts of the male reproductive system. So the vas deferens continue and will enlarge, so its contents will be transported into the ampulla of the vas deferens, which is enlarged part of the vas deferens. So this is the ampulla, and then it will empty the contents into the ejaculatory duct. Now also there are two seminal vesicles which empty their secretions into the ejaculatory duct just before it enters the prostate, the body of the prostate gland, which also empties its content into the ejaculatory duct. And then all these secretions will be transported into the urethra, the internal urethra. There are some glands that also have secretions, such as the bulbourethral gland into the internal urethra. And as you can see, this is the passageway of the all the content from these organs into the exterior. This is a cross section of the seminiferous tubules where sperms are formed, and we will talk in more details later on about the structures and the different cell types of the seminiferous tubules and the process of sperm formation. As the main function of the male reproductive system is to produce the sperms, which are the gametes that will fertilize the female gamete, the ovum, in order to produce the fertilized ovum that is the will produce the offspring. This process is called spermatogenesis, formation of sperms from immature cells called spermatogonia. Spermatogonia, as you can see here from this cross-section of the seminiferous tubules, lie in the inner lining of the seminiferous tubule, and actually they originate from the primordial germ cells that migrate into the seminiferous tubules during the 5th week of gestation. The production of sperms only starts at puberty. إذن في وقت أو عمر البلوغ تبدأ عملية تكوين السبيرمز، ولكن الخلايا اللي هم السبرماتوغونيا are present since birth، موجودين، لكن sperm formation will only start after puberty. And actually this process may decrease in older people and may cease in the age that is called climacteric, which is can also be present in females, and we will talk later about the female's reproductive system. There are types of cells that are supporting this process اللي هم النوع الأول Sertoli cells. Sertoli cells, if you look at the lower figure, are large cells, with envelopes of the cytoplasm, and actually they surround and support the developing spermatogonia during the process of making sperms or converting to sperms. So these cells actually are present in the lumen of the seminiferous tubule, and they provide nourishment and support for the developing spermatogonia. These actually start working under the influence of the follicle stimulating hormone of the anterior pituitary gland. Now the second type of cells are the Leydig cells. Leydig cells, you can see them in the upper figure, the view of the upper figure. These are cells present in the interstitium between the seminiferous tubules. The interstitium of the seminiferous tubules, and those cells actually are activated or stimulated by the luteinizing hormone from the anterior pituitary gland, and their main function is to produce and secrete testosterone, the main male sex hormone. Actually, the Leydig cells might be seen in the newborn of male infants only for the first few months of life, and after that they will disappear until the age of puberty, when the gonadotropic hormone is stimulated and when testosterone starts to be released under the influence of the luteinizing hormone, then we will see the Leydig cells. And then they will continue secreting testosterone until or throughout the whole adult life. Now, the hormonal factors that are important for spermatogenesis are first the testosterone, which is released by the Leydig cells in the interstitium. This hormone is important for growth and division of the germinal cells. Okay, so it's very important for spermatogenesis. Also, it has other functions that we discuss later. The second, the luteinizing hormone, is very important for spermatogenesis because it stimulates the Leydig cells. It has receptors on the Leydig cells and then will induce testosterone production and secretion. The third hormone is the follicle stimulating hormone, FSH, which is stimulated by the gonadotropic hormone from the hypothalamus. It stimulates specifically the Sertoli cells, and without this stimulation, spermatogenesis will not take place. So it is essential for spermatogenesis. Now, fourth, estrogen. There has been evidence that estrogens are released from Sertoli cells and synthesized from testosterone, so they might be probably be essential for the spermatogenesis process, and both, of course, are stimulated by FSH. Finally, the growth hormone. Growth hormone is important for the metabolic function of the testis. So it provides a background of growth that promotes many levels of the spermatogenesis, starting from the early division. And this was actually indicated when there is a lack of the pituitary growth hormone in cases of pituitary dwarfs. It has been noticed that spermatogenesis will be lacking. So without the growth hormone, or with a deficiency in the growth hormone, there will be no spermatogenesis. This is a schematic presenting the regulation of the function of the gonads and the different cell types responsible for supporting spermatogenesis. As you can see here, gonadotropic releasing hormone is secreted from the hypothalamus, which has a stimulatory effect on the anterior pituitary gland to secrete both follicle stimulating hormone and luteinizing hormone. As you can see here, Sertoli cells have receptors for follicle stimulating hormone, and the Leydig cells have receptors for luteinizing hormone. So luteinizing hormone will stimulate Leydig cells to produce testosterone. Also, follicle stimulating hormone will induce Sertoli cells to support and induce the process of spermatogenesis. Now, testosterone also play an important role together with the follicle stimulating hormone in supporting and stimulating spermatogenesis. Testosterone has also another effects, which are the androgenic effects that includes the male reproductive organs growth and the primary and secondary characteristics of males. Testosterone has a negative feedback on the anterior pituitary. When there is increased testosterone secretion, this will have a negative feedback on the anterior pituitary secretion of LH and FSH. Also, testosterone has a negative feedback on the gonadotropic releasing hormone. There is another inhibiting factor released from Sertoli cells. When there is increased induction of spermatogenesis, inhibin will be released from Sertoli cells and will have a negative feedback on the anterior pituitary secretion of LH and FSH. So let's talk about the process of sperm formation. The sperm-forming cells, which are the spermatogonia, they originate from the primordial germ cells. So in the embryogenesis, these cells migrate from the abdomen into the testis and line the seminiferous tubule as spermatogonia, which are immature. They line the seminiferous tubules, and until puberty, there will be no further division, or there will be no further development or change on the spermatogonia. But after puberty, which occurs around 13 years old, the gonadotropic releasing hormone will be stimulated, will be increased, and then spermatogonia will start mitosis, proliferating, and this process takes about 25 days to give rise to primary spermatocytes. So these primary spermatocytes have the diploid, the genetic material, about 46 chromosome, will enter the meiotic division, the first stage, and then will give rise to secondary spermatocytes, which are also diploid. They have 46 chromosome each. This process takes about 19 days, and then they will enter the meiotic division, the second stage, and that will give rise to haploid spermatids, which contain 23 chromosomes each. And these spermatids will further differentiate in a process that takes about 21 days into mature sperms. So one primary spermatocyte will give rise to 4 mature sperms. 2 of them has the 6 chromosome X, and 2 of them has the 6 chromosome Y. So if the X sperm fertilized an ovum, it will give rise to a female offspring, and if the Y carrying chromosome sperm fertilized the ovum, will give rise to a male offspring. The total length of spermatogenesis, starting from the spermatogonia into primary spermatocyte, and then secondary spermatocyte, and finally into mature sperms, will take about 74 days. Now, the characteristics of the mature sperm is that they are composed of head and tail, which is the flagellum. And mature sperms are capable of movement, are motile, and also they are capable to fertilize the ovum. They need to be in a neutral or slightly alkaline medium, cause highly acidic medium can cause death to these sperms or shorten their lifespan. Sperms that are ejaculated from the male reproductive organs can live in the female genital tract only for not more than 2 days. The head of the sperm contains a structure called acrosome, which is like a cap surrounding the surface of the head of the sperm. It contains large quantities of the enzyme hyaluronidase and also the proteolytic enzymes. And these enzymes are necessary to digest the cells that surround the ovum in order to facilitate the fertilization process. Also, the tail contains neck and body structures. They contain mitochondria as well as microtubules, which microtubules facilitate the movement, the sliding movement that allows a back and forth movement of the flagellum or the tail of the sperm, which facilitates the movement or motility of the sperm. After formation of the sperms in the seminiferous tubules, they have to pass through the epididymis, and at this point they are not capable of motility, but within about 2 days they develop the capability of motility in the epididymis. إذا الآن بيصيروا modified بحيث ممكن إنهم يكونوا capable of motility. بالرغم من ذلك they will not be motile at this point. Why? Because there will be inhibitory proteins in the epididymal fluids that prevent their motility, but they are capable of motility but inhibited from motility until after ejaculation. Okay. Average sperms formation in the two testes of adult human range up to 120 million sperms each day, and actually small amounts of this number will be stored in the epididymis. The rest will be stored in the vas deferens and up to 1 month. So the life expectancy of the sperms formed, mature formed sperms are only 1 month. After ejaculation, the sperms they become motile because they are capable, and the inhibitory factors will be removed or washed away, and then they will be capable of fertilizing the ovum in the female genitalia. And after undergoing the process, completing the process of maturation. Also, the sperms are provided with fluids full of nutrients secreted from Sertoli cells and the epithelium of epididymis. And these secretions contain testosterone, estrogen, and a lot of enzymes and nutrients essential for the process of sperm maturation. Other than the fluid containing sperms from the testis and the epididymis, there are other fluids secreted from different male glands, which are the seminal vesicles, the prostate gland, and the bulbourethral gland. And the secretions from the seminal vesicles comprise about 70% of the total volume of semen, and it's composed of a mucoid material which gives the viscosity of the semen. Also contains nutrients such as fructose, as well as the prostaglandins. It is acidic because of the citric acid, and it contains fibrinogen that is necessary for coagulation, and coagulation aids in the movement of sperms and semen in the female genitalia. The prostaglandins help fertilization by either reacting with the female cervical mucus, making it easier for the sperm to move, and second, by facilitating the peristaltic contractions in the uterus and fallopian tubes, which also enhance movement of the sperm towards the ovaries. And this is actually very important for the fertilization to take place. Third, it can also, it has been suggested that it can reduce the immune response in the female reproductive system toward the foreign sperm. Now, the prostate gland also have secretions that are characterized by being thin, milky, and alkaline. In the pH, they contain calcium and citrate as well as phosphate ions. Also, it contains clotting enzymes and profibrinolysin, which is a fibrinolysis enzyme. The alkaline fluid is important to neutralize the acidic fluid that comes from the vas deferens. And as we know, acidic media is bad for the life of the sperm, so it reduces the life expectancy of the sperms and inhibits fertility. Second, to neutralize the acidic vaginal secretions in the female reproductive system, which is also a bad environment for the sperm. And so it promotes neutralizing this acidity will promote the life span of the sperm to facilitate its viability and then fertilizing the ovum. So in summary, semen that is ejaculated is composed from fluids and sperms that is coming from the testis and epididymis through the vas deferens, which is only 10% of the volume. And the majority is coming from the seminal vesicles, which comprises about 60 to 70%, and 30% of the semen is coming from the prostate gland alkaline secretions, and very small amount comes from the mucus gland and our mucus secretions from the bulbourethral glands, which facilitates the movement and lubricate the movement of secreted fluids. In the female genital tract, freshly ejaculated semen undergo a process called capacitation, which take place within 1 to 10 hours after being expelled or ejected. First process of this capacitation is washing away the inhibitory factors that inhibit motility of the sperms by the uterine and fallopian fluids. Second, the sperm will swim away from cholesterol vesicles that covers the acrosome in the head of the sperm, so the acrosome gets thinner. Third, the membrane of the sperm becomes more permeable to calcium ions, which is necessary for the fertilizing the ovum process. The main sex hormone of the male reproductive system is testosterone. Although the testis secretes several male sex hormones which are collectively called androgens. إذا androgens هم كل الهرمونات المتعلقة بالصفات الذكورية تسمى الاندروجينز اللي هم testosterone, dihydrotestosterone and androstenedione. So the main hormone of all of these is the testosterone, which is more abundant. However, the less abundant and more potent hormone is the dihydrotestosterone, which is the more active form. But most of the testosterone is converted into dihydrotestosterone in the target cells. So the term androgens are related to the masculinizing effects of these hormones, which are important to give the men or the male secondary characteristics, physical characteristics. And testosterone is secreted from the testis, and androgens can be secreted from both testis and the adrenal gland. So the origin or the source of these androgens is either from cholesterol or directly from the acetyl coenzyme A. The intracellular mechanism of the testosterone action is that testosterone, as we know, is a steroid hormone, so it is capable of entering or diffusing into the target cell. Then it will be converted by 5-alpha reductase into dihydrotestosterone. This will combine with cytosolic receptors, androgen receptors, and then will be translocated as dimers into the hormone response element to induce gene expression that ends up with increased protein synthesis in the target cells. So it is considered an anabolic hormone. Testosterone is not only secreted after puberty in adult males in order to induce the process of spermatogenesis to produce sperms or to enhance the appearance of the male characteristics. But also testosterone is secreted in the fetal and neonatal life. As we can see here from this graph, in the middle trimester of gestation, there will be a peak, an increase in the plasma level of testosterone that is primarily induced by the hormone human chorionic gonadotropin hormone, which has similar effects to luteinizing hormone in secreting testosterone, and also in the fetus and the neonatal life, about 10 weeks after birth, testosterone levels will also be high without proceeding into sperm production. And this is actually responsible for the male reproductive organs growth. The main functions of testosterone released during fetal and neonatal life is that testosterone that is secreted by the genital ridges in the first 7 weeks of gestation and the testosterone secreted in the last trimester is responsible for development of the male body characteristics, including formation of penis and scrotum. Also, it suppresses the formation of female genital organs. So, in the absence of testosterone, a male offspring will develop female genital organs instead of male genital organs. Also, testosterone is important for descending the testes, which usually occur in the last 2 or 3 months of gestation. Testosterone and increased secretion of testosterone is responsible for this process. And if failure occurred of this process, non-surgical treatment of a baby can be injecting testosterone in order to help the testes to descend into the scrotum. The effects of testosterone in the adult life and after puberty is the appearance of primary and secondary sexual characteristics. So after puberty, testosterone release and increased amount of testosterone will cause enlargement of the male sex organs, as well as the appearance of secondary sexual characteristics of males. Also, it affects the distribution of body hair, so it increases the growth of hair over different areas of the body, including the face, abdomen, and also for some people it increases the chance of having baldness on the top of the head if the genetic background was present, the inheritance of the baldness characteristic, which is if combined with large quantities of androgenic hormones such as testosterone, baldness will appear. Also, testosterone has an effect on the voice. It causes hypertrophy of the mucosa in the larynx, as well as larynx enlargement, which will cause a cracking of the voice and then will give the masculine voice characteristic of adult males. Testosterone affects the thickness of the skin and also increases the sebaceous gland secretions, which may contribute to the development of acne during this stage. Testosterone also increases the protein formation, which is the anabolic action of testosterone, as well as muscle development, which is a masculine characteristic of males. Testosterone has an activity on the deposition of bone matrix, increases bone matrix and calcium retention in bone, which increases the thickness and the strength and the quality of bone, which is also due to the anabolic effect of testosterone. Also, it increases the basal metabolic rates of the body about 15 to 20%. And it has a positive effect on increasing red blood cells. It increases red blood cells up to 20%, and it is believed to be due to the increased metabolic rate as a general effect of testosterone, and it's not related to erythropoietin levels. Also, it has an effect on increasing electrolyte and water retention, as many other steroids, similar to the or lesser extent to mineral corticosteroids. So it increases reabsorption of sodium in the nephrons of the kidney. Now we will go very quick through some of the abnormalities of spermatogenesis and male reproduction function, just to emphasize on the roles of the components of male reproductive. Sperms count as well as shape of sperms affect fertility, الخصوبة. The quantity or volume of semen that is ejaculated is about 3 to 5 ml, and in every ml there will be an average of 120 million sperm. That's the normal average of a sperm count. The effect of sperm morphology and motility also is very important. So sometimes the sperm count can be normal, but the shape or the motility is abnormal, which will cause infertility. And these are some of the shapes that are abnormal, and they might cause infertility. So even if the shape of the sperm is normal, but the motility is affected, if it's non-motile or of motility is low, this also can cause infertility. Some of the prostate gland abnormalities are the benign prostatic fibroadenoma, which occurs in older ages due to over proliferation and growth of prostate tissue. And this condition is actually not caused by testosterone, it's independent of testosterone. However, cancer of the prostate gland is caused by stimulation by testosterone of the cancerous cells. Hypogonadism in males can take place in different stages of male development. If hypogonadism was during fetal life due to non-functional or absent testes, there will be no male sexual characteristics that will develop in the fetus. And instead, there will be female organs development. However, if the body loses the testis before puberty, this will cause a condition called eunuchism, which is characterized by having infant sex organs and infant sexual characteristics. And if the man has been castrated, removed his testis after puberty, then sexual organs may regress in size and voice may regress, but the main male secondary characteristics will remain. Hypogonadism can be due to genetic inability of the hypothalamus to secrete a gonadotropic releasing hormone, which can be resulting from the hypothalamus, from the central nervous system. And this actually is accompanied with obesity as well as eunuchism. And this is a syndrome called adipose genital syndrome. Cryptorchidism is the failure of the testes to descend into the scrotum, which takes place during the fetal life. Testicular tumors and hypergonadism in males, if happened due to interstitial Leydig cells tumors, which are very rare, there will be overproduction of testosterone. And if it took place in children, this will cause rapid growth of the musculature, bones, and also excessive development of male sexual organs. But if it took place in adults, it might be hard to see or to recognize the effects of the high testosterone secretion from the tumor. Now, there is an assignment for you to self-study the last topic in male reproductive system, which is the male sexual act. And I presented a slide summarizing this topic, and the textbook for you is Guyton and Hall, and I will provide you with the syllabus of the suggested readings from the textbook for the purpose of examinations. Thank you and good luck.